Thank you, Sharon. Our final speaker is Kenneth Offit. And Dr. Offit is Chief of the Clinical Genetics Service at MSKCC. And he's entitled his talk, A Tale of Two Genomes, Current Generation Approaches to Cancer Susceptibility. Dr. Offit.
Thank you very much. I apologize for the old-fashioned adenovirus that I seem to have picked up. But if you bear with me, I wanted to also just acknowledge the sort of historical significance of the occasion. John Mulvihill kindly sent around some old 35 millimeters that we scanned in, and so we actually, I had to cheat a little bit to get them all in the same. You can see, I think Joe was taking the picture, I was told. But all of these individuals I had the opportunity to meet as visiting scientific consultants at Sloan Kettering, and I was a young faculty member. And unlike some where Joe had been encouraged to leave, they encouraged me to stay and said, stick it out. We know that it's all tumor genomics, but it'll all come around in the end, you'll see. And this point of the synergy, we understand that our positional cloning of genes and families gave us RET, NF1, APC, BRCA, genes that we're working on in leukemias and lymphomas. And I've chosen the point in this talk. I'm going to tell you three stories. I'm going to stay in hematopoietic malignancies because this is really where the somatic genetics began in our collective scientific lifespans. We're going to talk about the understanding of shared pathways of germline and somatic genomics that is needed to inform pleiotropy observed for susceptibility to lymphoid leukemia and lymphoma. And that's the major topic that I'll be covering today with a major emphasis on transcription factors. I'm also going to give you some new data at the end. This will be a mixture of published and a fair amount of new data with some provocative data on childhood cancers and de novo mutation rates. So the evidence for familiarity of leukemia and lymphoma comes from the twin studies, the Scandinavian studies of 45,000, recently updated to over 150,000 twin pairs where leukemias and score pretty high, highs relative to, say, breast and colon cancer. The sibling studies show this rather skewed number for acute lymphoblastic leukemia in children. But of course, if you have twins and you have contamination through the placenta and the same immune systems, well, that's what that's all about. So you can't really use identical twin data for ALL. But you can use the SIB data, and you can see there that the relative risks are still significantly increased, and lymphoma as well. And in the Utah genealogies, where you can calculate how related everybody is to everybody else in a hospital ward or anywhere else, you can see leukemia and lymphoma right up there with breast cancer. So these are significant public health. If you take together all leukemia, lymphomas, myelodysplasias, and myelomas, it's about 170,000 cases a year. Breast cancer is about 220,000. So it's significant for public health and significant also when one is speaking about the familiarity. This is what I was doing when I had the opportunity to meet with Joe, Bob Miller, and Louise during those early years. I was describing chromosomal translocations associated with leukemias and lymphomas. The last authors of these papers, I was the youngster, were R.S.K. Shiganti and Ricardo Della Favra. And these descriptions that we did at this time led to the cloning of PAX5, BCL6, and BLIMP1, better known as PRDM1, over the course of the next few years. What we observed, and I spent some time describing, was the pleomorphic presentation. MYC described in Burkitt's lymphoma, but also large cell lymphoma. BCL2, classically in follicular, but also in large cell lymphoma. PAX5, I described it in a lymphoplasmacytoid leukemia, but also we'll be talking about it in acute leukemia. BCL6, also in follicular and diffuse lymphoma, and a lot of work there. And then finally, the PRDM1. Interesting gene, transcriptional repressor, described originally in 6q. I was mapping 6q deletions, and Ricardo then went on and identified this transcriptional repressor seen in a variety of lymphoid and also solid tumors. At the time that I met with that distinguished crew, I was collecting families like this, where you had Hodgkin's disease, large cell lymphoma. We even had leukemias, ALLs. So I was convinced early on, and this is 20 years before we actually got to do any genomics, that there was a mixed lymphoid susceptibility syndrome. And that's what we'll be describing. Now, there was one other guy at Sloan Kettering who was interested in this before my time, Henry Diamond. He was interested in it because he was diagnosed with Hodgkin's disease when he was in the Navy, came to Memorial, and then was treated at Memorial, and then actually became an oncologist, joined the staff at Memorial, and became the head of the lymphoma service, while over 17 years he was being treated for Hodgkin's disease. Incredible story, right? Had surgeries done every time and radiation therapy, kept him going, Dr. Diamond, for 17 years. And toward the end of his life, just as chemotherapy was coming in, this character, Mort Lacker, shown at the top, was bringing in chemotherapy. And this is my story. So Mort wrote that Joe Fraumeni was a chief resident in the Department of Medicine. I was a newly appointed member of the lymphoma service. Dr. Diamond was in a private room on the 6th floor of the hospital. I spoke to Dr. Fraumeni, told him that although it was probably much too late to get any effect from IV chemotherapy, it was worth a try. In the middle of the night, Dr. Fraumeni and I went into Dr. Diamond's room and gave a single dose of IV Theratepa. So another first, okay, for Dr. Fraumeni, that unwittingly, okay, participated perhaps in one of the early uses of IV chemotherapy in Hodgkin's disease. I'm sure that this was done appropriately, with IRB and informed consent. I want to make sure that, we absolutely, make that clear. But it is an extraordinary accomplishment. Now, one of the ironies, of course, is this is a readily curable malignancy. And in fact, our major concern with Hodgkin's disease are the second cancers that you get after Hodgkin's disease. And so as we switch now to the modern era of genome-wide association studies, it's remarkable that we were able to do a study with 62 cases of secondary cancers in patients who had had Hodgkin's disease and 71 controls. This was done with Ken O'Neill, one of my postdoctoral trainees, and I told Ken, you can't do this study. You can't do a GWAS study with these numbers. And yet we had a 10 to the minus 8, which of course got this into Nature Medicine. But interestingly, look at the candidate gene, PRDM1, that same 6q minus deletion mapping gene that we'd identified 20 years before, lost in some solid tumors. Functionally, we showed that if you looked at cells that are homozygous for the protective haplotype, you could see that these were induced after radiation therapy. And also you could see that in cells where PRDM was not induced by radiation therapy in cells homozygous for the risk haplotypes. This is the risk haplotype, and you can see the lack of induction here. So this is a functional demonstration of the significance. Not all GWAS studies are anonymous and in intronic regions. That's the theme I'll be mentioning. As we switch to CLL, this is our genome-wide association study of the lymphoid family. So we took our 280 families that had multiple types of lymphoma and actually put them together and did this genome-wide association study. And the major hit that came out of this is in the HLA region, which is a common theme in the lymphoma genome-wide association studies. And I'm going to come back and speculate a little bit about the functional biology of that as we go along. In CLL, the genome-wide association study was done as part of the Interlymph Consortium. I'm going to mention a little bit about that in the next slide. This was a very large study of 2,000 CLL cases and controls. But again, I'm trying to bring together here germline and somatic. That's the theme of this talk. In the genome-wide association studies in lymphoma and leukemia. I've spent most of my life in those 20 years doing breast cancer, but in the hematopoietic malignancies, it's very clear how these relate. FAS mutation, FAS associated SNPs were the major hit in the CLL cases. And you can see those appearing here, as well as SNPs in the regions of the breakpoint of BCL2, which is in the BCL2 translocations that we described, with clear biological correlates that come to mind. But the problem with these is that the types of biology, and Stephen Chanek has set this up very nicely, but most places are not able to move back and forth between the SNP work and the functional work that's required. As an example of this, 10 years earlier, we had a family like this with 3 cases of hairy cell leukemia. If you can see this incredible pedigree. I remember this family because he came in and I wanted to get his blood, so my genetic counselor drove his car around the block because he couldn't get a parking spot while we drew his blood. That's how we have to do research in New York. I'll always remember this case. We made cell lines out of this. And we found is that if we actually gave an anti-FAS antibody in the cell lines and measured apoptosis by an exon 5 binding, you could see in this family, number 2, the same abnormal apoptosis as one would see in a hereditary syndrome of a total knockout of this gene, which is the Canal-Smith syndrome. We showed there was normal FAS expression, and this is a year of work done with several collaborators, failure to activate caspase 3 and decrease caspase 8 flip protein expression ratio, and wrote this paper. But that was in 2003. To get all those cell lines up and to get them running again, which we will do, now that we know that FAS is actually the operant site in susceptibility to CLL, is a challenge. This is whole exome sequencing of a kindred with CLL, and again, this is unpublished data, but to point out that the truncating mutation that we found in PN13 is a protein tyrosine kinase phosphatase, is exactly at the region of interaction with FAS. So you're seeing a commonality here of themes. And this will be work done as part of a funded R01 by my postdoc, and these are the experiments now that will take place over the next year. As we look at the large lymphoma association studies, and here's where I want to take my hat off to the NCI DCEG intramural, this program. So these studies, unlike, I will say, the collaborations I've been involved with the UK group, which tend to be centric on the Greenwich Mean Time, and that sort of place in Cambridge. Here, what's happened is that the NCI, and hats off to Stephen Chanek and Nat Rothman, this has been truly collaborative, and the meetings bring in young investigators. And although I can take some claim here because my first author is a shared first author on all of these papers, so I have all of the data and we've been able to participate, this has been a true joint collaborative effort, with the data publicly deposited, by the way, also unlike the Greenwich Mean Time experiment. So I'm not going to talk about this different SNPs because most of this work is still in various levels of submission, resubmission, and revision for Nature Genetics. But I did want to sort of give a sort of my meta-analysis, with apologies to Steve and Nat if you're in the room, but this is what happens when you let data sit, that we can analyze. And of course, all of this work has been done by Vijay Joseph in my group, who's on weekly teleconferences with the NCI and other collaborators. First is the HLA overrepresentation, which you see here in follicular lymphoma, large cell lymphoma not as much. So this extraordinary role of HLA. And then if you wanted to probe a little bit, we now have the ENCODE data set, so you can look at these SNPs and get an idea of the functional readout. The ENCODE data set is set up a number of ways, but one of the annotations has to do with with functionality. And so if you do that, and here's a lymphoblastoid cell line, which is the only one relevant here, and the red is showing promoter region activity, that there is a strong evidence, at least when one is looking at large cell lymphoma, that these SNPs are involved in active regions of transcriptional activation as read through the ENCODE. Another way to look at this, which Robert Klein has done in our laboratory, has been to look at the DNase marks in ENCODE. And again, you can see this shift that all of these lymphoma GWAS SNPs are overrepresented when one is looking for regions of transcriptional activation sites, giving you an idea of the important biological pathways that one is dealing with. The HLA story is one that fascinates me. This is a supplementary figure in one of the large cell lymphoma papers, pointing out that many of the SNPs that are identified in the follicular lymphoma paper are involved with the coding of the proteins that are sitting right at the binding site of the binding groove of HLA, the peptide binding groove. These SNPs are imputed on the basis of strong LD, and they can predict strong protein function. Chris Sanders has been a leader at this in this notion of using SNPs to actually impute protein structure, and here this kind of work is happening in the setting of a lymphoma GWAS. getting to this issue of the association with HLA. Now, what is this? I mean, marginal zone lymphoma, there's clearly an inflammatory relationship in the epidemiology of lymphomas. What the association is with HLA, which is so robust, is, I think, an enormously fascinating challenge that needs to be deciphered. And then to sort of one-up Malkin, this data I got on the plane, okay, this morning from my postdoc, okay? And apologies to Chaddock. This is actually a pathway analysis, which is just pointing, and this is on the data from the 4 GWAS studies in the interlymph that are in various levels right now of communication, just telling them what I think they probably knew, that these pathways in apoptosis are overrepresented, FAS, caspase, and BCL2. The other story that I wanted to just tell you is one relating to acute lymphoblastic lymphoma. This is one of the experiments that has worked very nicely, where we had one family. We were able to ascertain all of these individuals. It's quite a family. If you look at this, this woman is a nurse, came to us because she had 2 kids with leukemia, one of whom she lost. It's not a nice thing to lose a child with leukemia nowadays, anytime. Multiple CNS relapses, and now you can see here that now her husband's sister is married multiple times, and in each of these marriages, on 2 of them, he has a child with leukemia. So it's quite an extraordinary family saga. So we had DNA from all of these individuals. We did the subtraction to figure out what is it that the individuals who were affected had that the mom didn't, because the mom is married in, so we used her as the control. And we were able here to zero in on a PAX5 missense mutation as the one of interest. Found another kindred with this same exact mutation at St. Jude's. And working with Charles Mulligan, we were able to put this together. This mutation is an oncopeptide binding region. It's in a conserved region. Looking at the extraordinary data that we've been able to put together with St. Jude, we could, even in a sort of agnostic look at RNA-seq, profile out these hereditary leukemias just by looking at the PAX5 binding partners. But to be more specific, since we know a lot about this transcription factor, we were able to show that the transcriptional activity of these variants compared to wild type determined in a luciferase reporter assay was clearly, you can see there's the glycine 183 serine, here's our case, and here's the control, and these would be nulls. And then in another readout, one of the things PAX5 induces is the production of IgM on the surface of cells. We put it in a cell line that doesn't have PAX5, and we're able to show, again, this sort of hypomorphic effect of the mutant here against the wild type. So that was a nice story, and we've been able to go on with that to find another family now in Israel, a Jewish family. Maybe a founder mutation. You heard Sharon. Not everyone that's in a Jewish person is a founder mutation, okay? We screened hundreds and hundreds of cases, and this is null. So interestingly, it's a common haplotype, and this same mutation is occurring in different ethnic backgrounds. It's not a founder mutation. Here's whole genome sequencing of a familial ALL. This one we've just done recently. So this is genomes now, not exomes. And what's interesting here is that if you do this with exomes, you'll see all of the SNVs. And this is not a very pretty picture, is it? But if you have genomes, you have the opportunity to just sort of limit yourself to structural abnormalities. And then all of a sudden it's like, well, that you can see. And so we have here, we think, a good candidate identified through genomes that we would not have picked up through exomes. And we'll be characterizing that further. Intan Schroeder, an MD PhD in the lab, has been studying in collaboration with colleagues from Australia another large family with ALL. Here we've gone back to old-fashioned haplotyping to help us feel our way in the dark. As you can see, the family structure here is actually sufficiently informative to give us that, enough meioses between individuals, and allowing us here to zero in on a candidate region in this family as well. So these are unpublished data that are, I think, indicative of sort of an exciting new time in next-gen sequencing and showing us mechanisms of inherited susceptibility. We've been working with Charles Mulligan extensively over the last 6 months. This is an appendix in one of Charles's papers in Nature Genetics. This is Jim Downing presented an updated version of this at AACR. What's striking about this is in the germline of the data set of childhood leukemia is a remarkable number of deleterious mutations of putative cancer susceptibility genes. And I think what this data begs is for us to expand these families and analyze and to see if, in fact, there's a much larger proportion of pediatric ALL and pediatric cancer in general due to hereditary predisposition. We have an R01 on this that will be resubmitting, reasonably scored, and we're hoping to continue to get at this question in collaboration with our St. Jude's colleagues. The third story, and this is one which is potentially will allow another answer to the big question of the missing heritability. And this is true in breast cancer, it's true in lymphoma, where only about 15 to 20% of the heritability is explained by the GWAS studies and these rare variants. What are we missing? So we set out to do this a couple of years ago with Mike Wigler at Cold Spring Harbor, where we're taking trios, so unaffected parents and then kids, although they weren't all kids. I mean, you'll see we had a lot of young breast cancer cases and colon cancer cases, to look, and we were doing this with exomes. We did copy number variations initially and found a 7% CNV pickup in testicular germ cell tumors and published that, but then switched to exomes. And what we're finding is that there are a significant proportion of loss of function mutations. In this first group that we've done, 4 of 17 of these cases, trios, and these are well genotyped, are showing loss of function mutations in the kids and not in the parents. So these are de novo mutation rates. Now, this is not surprising to those of us in the clinic. I mean, we know, for RET mutations and RB mutations, there's significant de novo mutation rates. Just BRCA, we don't seem to have any real de novo mutation rates, although we have also documented those. This, it's provocative data, and I showed this actually at a retreat, and Eric Lander was in the back, because there's a paradox. These kids historically would not have reproduced, and this is why you could maintain a de novo rate and yet still see overall the rates of germline mutations that you see, for example, in prevalent screens. So it's an interesting hypothesis. I'm mentioning this in this setting because we have one in ALL, which looks real, de novo nonsense mutation in an interesting gene, and we're in the process of working this up. It has validated. And of course, each of these has to be functionally also described in order to be maximally convincing. So I'll just end on this broad note, from pedigree to population. Sharon showed you the story, the ARTEL collaboration, and I will just emphasize what she said as a clinician. It's a big deal, to have a carrier mutation frequency that you can screen and prevent cancer. But I would also go one step further and say that other than that intervention, we can do pre-implantation interventions as well. And the pre-implantation genetics is something that I wrote about early on. It's something that we put into the practice of what we do, and it's something that we tend to forget about. The earliest case of this actually when we first started discussing this was in a P53 setting and offering pre-implantation genetics. And this is a family with Lee Fraumeni. And I've since counseled, medical students with Lee Fraumeni. It's an incredibly challenging clinical interaction, as I think you heard from the presentations earlier. But one thing's for sure is that these families can reproduce without passing on these mutations. So this ALL family that we published and that I just described to you, I had the honor or pleasure, to speak to that woman, the nurse who came to us and say that your family will never have another leukemia in it because we make this a CLIA test and we can do pre-implantation genetics. And this is quite a miraculous intervention that's available right now. So we will make significant strides even in the absence of major functional advances in the understanding of the genomics. So I'll conclude by saying that germline cancer genetic information informs tumor genomics and vice versa. Current generation sequencing has resulted in a collision of cancer and germline genetics, and that is because as we sequence tumors, which we are now doing at all of our major cancer centers, we are also sequencing the germline, and this has created an enormous challenge in the profession of oncology. The repertoire of functional biology used to probe tumor biology now is required to elucidate functional significance of these germline variants, a term which is quaintly termed curation by my colleagues at NHGRI. We call this cancer genomics. The burden of de novo mutations is higher than expected in pediatric cancers, it appears, from some of the work, and that in the classical, the best of times and worst of times, 2 genomes, 2 cities. It's definitely the best of times with regard to the prospect for utilizing pedigree-based genomics for cancer prevention. Thanks.